Where's Brad? Are we being streamed to the other campus? Okay. Well, wow, this is what happens when you have a speaker like young Dan come. The place gets packed very quickly and everybody's here on time. This is usually everyone drags in at 4 minutes after the hour. And so this is a novelty. In fact, it's a novelty that Mike Fox is the last to come into the room. So it's a pleasure to have her here. It's been a pleasure, I think, for everybody that she's talked to talk to her. She's really just an amazing scientist and an example to all of us, in particular the young people here. That does not include me anymore. She got her BS degree in physics from Beijing University in 1988 and her Neurobiology Ph.D. at Columbia University in 94. I'm doing this off the paper so I don't make mistakes. It's an extremely impressive CV, and it's really, really thin. Both of those things are true because she just says, here's who I am, and here are the papers I've published. It's very different than what you see typically in medical arenas, but I'll mention a few things.
That's important, so next time I'll write a longer CV.
Very, very fat, very, very fat. In fact, you could put blank pages and stuff them in. No. In 2009, she became a Howard Hughes medical investigator. She was started as an assistant professor at Berkeley in 97, rose very, very quickly through the ranks and became a full professor in, 2005. And she's in molecular cell biology department. One of the things that characterizes her work is that she's worked on not just one thing. And by that I mean she started in the visual system, and not just in the visual system. She worked on vision and elements of how does the visual system process information related to synaptic plasticity, spike timing dependent synaptic plasticity, modeling, et cetera, and was a leader in that area up until, I guess, about a decade ago. And she decided to switch gears and move into another fundamental area, which is sleep. And what we're looking at here is really a culmination. There's a paper out this year, 2020, in Science, which is just a tour de force. I was talking to her last night asking how many postdoc lives were sacrificed in the making of that paper. And apparently it's the first author that was the main workhorse in that. But I recommend looking at it because of the range of kinds of techniques and questions asked and the rigor involved is kind of stunning, really. So with that, I'd like us to welcome Dr. Dan to Virginia Tech. Thanks.
Thank you so much, Reed, for inviting me. I've had a lot of fun talking to people and learning the things that I didn't know much about, which is very different from what I'm going to be talking about, which is sleep-wake control, something that we started working on about 9 years ago. So sleep is an essential innate behavior. It's innate because we know how to do it when we're born, right? No learning is required. In fact, the little babies, they sleep much better than most of us in this room, right? That's why we want to sleep like babies, right? So it's essential because as far as we can tell, all animals sleep. And that includes flies and worms and even jellyfish. They don't even have a central nervous system. They have a very primitive kind of neural net. And recently people show that they even have some kind of sleep behavior. So for non-mammalian animals, we usually detect their sleep by a lack of movement, right? So they stay still for a while. For flies, if they still, stay still for like over some number of seconds, we score that as a sleep episode, right? And if you give them some sensory stimulation, they show no motor response. So basically, it's detected by movement. Now, for mammalian animal models, we measure EEG and EMG. Oh, I'm sorry, I didn't. Oh, sorry. Okay, so now you can hear me, right? Yeah. So for mammalian animal models, we measure EEG and EMG. This is just a standard in the field. So during sleep, there's a very clear reduction of the EMG power, which indicates muscle tone. And based on EEG, we can also tell the difference between non-REM sleep and REM sleep. So REM sleep is rapid eye movement sleep, and that's when we have vivid dreams. So for humans, if you're brought to a sleep clinic, we measure EEG and EMG and also things like heart rate and breathing and some other things. So when we fall asleep, basically we shut down somatic movement, motor activity, which is reflected by EMG. We also slow down a lot these autonomic motor activity, right? Heart rate is down, breathing rate is slower. So the point is that falling asleep is not just about changing the brain state, which is reflected by the EEG. It's also about reducing these different types of movement. So my lab is interested in what controls sleep, at least for the past 9 years. And what I hope to convince you today is that a lot of the neurons controlling sleep that put us to sleep are really part of the motor circuits controlling these different types of movement. And that's why I had this weird title for my talk. So pioneered by Morosi and Magan, who discovered the so-called ascending arousal system, now we know that there are a lot of neuromodulatory systems important for arousal. So these include a bunch of monoamines like histamine, dopamine, serotonin, and what else? What did I say? Norepinephrine, right? There are also cholinergic systems in both the brainstem and the forebrain. Also some neuropeptide systems, and the best known one is called orexin/hypocretin, right? So if you lose that, you have narcolepsy. Now, in addition to these well-known neuromodulatory systems, there are also a lot of regular glutamatergic and GABAergic neurons in these subcortical regions that are important for arousal. So just to illustrate that point, and also to give you some concrete ideas about the kind of experiments that we do, I'm going to show you this video here. So basically, this little black thing is the mouse, right, in the home cage, and this is his nest. So this cable here contains the EEG and EMG recording cables and also an optic fiber so that we can optogenetically activate some cells in this mouse brain. And then you can look at the behavior. So when you see a blue square showing up here, that's when we turn on the laser. You can't really see the laser here because we actually wrap it around with black tape so that the mouse doesn't see the light. And then here, I'm showing the EEG power spectrogram, the EMG trace, and the color-coded brain state, right? So they're wake and non-REM and REM sleep. And so the laser is indicated here. So here the mouse is, oh, did I start? Okay, yeah. Okay, so the mouse now is asleep. The laser comes on and you see immediately he woke up, right? So even without looking at the EEG and EMG change, you can kind of see his behavior. So when he wakes up, the EEG is immediately desynchronized. You lose this low frequency activity. And again, laser comes on now, and you see that he wakes up immediately, right? So it turns out that, when I first saw this about 9 years ago, when we first started looking at sleep, this is far more robust of a behavior effect than any of the vision studies that we've done. So this is very exciting, right? So I used to stay really late at night in my office and just to watch these movies, right? But it turns out that this is really pretty common, right? So you don't even need to be very selective. You can almost randomly pick a population of cells somewhere in the hypothalamus, and you're going to turn on the light, and this is what you see. So the point is that it's actually very easy to find neurons in the brain that wakes the animal up, right? But it's much, much harder to find neurons that put us to sleep. So according to the textbook version, there's a single sleep center called the preoptic area, which I'll refer to as the POA, in the anterior part of the hypothalamus. The idea is that there's a subset of the POA neurons that promote sleep by inhibiting a lot of these wake-promoting neurons, the same neurons that are shown here. And indeed, using retrograde labeling from some of these wake neurons to back label the cells in the POA, we were able to identify some of the POA neurons that promote sleep. But this is far from the whole picture. About 4 or 5 years ago, several groups, including us, have found some neurons outside of the POA that also promote sleep. So all of a sudden, this idea of a single sleep center in the brain just seems to be wrong, right? Because we found some neurons here and they found some neurons there. And then to get an idea of what the entire sleep control network might look like, we decided to do a whole brain screening for sleep neurons. So we have two basic criteria for sleep neurons. One is that they should be sleep promoting, right? So their activation should increase sleep, their inactivation should decrease sleep. And the second one is that they should be sleep active, right? So they need to be active at the right time to do their job. And when we screen for sleep neurons, we also have two corresponding strategies. We can either screen for sleep promoting neurons and then see if they're also sleep active, or we can screen for sleep active neurons and see if they're also sleep promoting. So I'm going to give you an example of each approach today. So to screen for sleep active neurons, we use this technique called Fasttrack. So I'm going to get into some of the detailed technical stuff which you guys don't use, but just to give you a flavor of the kind of approach we can do. So anyway, so the Fasttrack technique was developed by Li Chunluo's lab at Stanford University. We crossed two mouse lines together. One of them expresses Cre-ER under the FOS promoter. So FOS is an immediate early gene, right? So the idea is that when a neuron is activated, they turn on their FOS gene. And so in this mouse line, they should also express Cre-ER. And then this other mouse line expresses GFP in a Cre-dependent manner. And I'll explain to you how this works, right? So in this crossed, so the good news is that both of these mouse lines you can buy from Jackson Lab. So we divided these cross mice into two groups. One of them we call them the SD mice. So we sleep deprive them for 6 hours, and we inject tamoxifen during sleep deprivation. So the idea is that during sleep deprivation, the neurons that are wake active, they should be active and express FOS and therefore Cre-ER. And tamoxifen should cause the Cre-ER to enter the nucleus, and that allows the activation of GFP to permanently label these putative wake active neurons. And then in this other group, we call them the RS group. So we first sleep deprive them for 6 hours to make them really sleepy. And then we allow them to go into sleep, right? So we stop harassing them. And then because they're so sleepy, they go into really solid sleep for at least 3 hours. And then we pair the injection of tamoxifen during the rebound sleep, RS, so that the Cre-ER that are active in the sleep neurons can enter the nucleus and cause the GFP labeling of the sleep neurons. And when we compared these two groups of mice, we found that some brain regions show more GFP-labeled cells in the RS group, these guys, and some other regions show more expression in the SD group. So if you look at this region, the top candidate with the highest relative expression of GFP in the RS group, right? So these are the putative sleep neurons. This region is shown here. So it's basically the ventral part of the periaqueductal gray, the PAG. So this is the aqueduct, and so this is the ventral part. So you can see that there are a lot more GFP-labeled neurons in the RS group than the SD group. And when we saw this region, because nobody was talking about this region in the sleep field, and we actually got very excited. And that's because in the sleep field, everybody knows about this guy, Konstantin von Economou. So he was the one who really popularized the idea that the POA is the sleep center. And he did that based on analysis of post-mortem human brains. You know, these people had sleep problems. But in fact, several decades before von Economou, this guy here, Ludwig Mausner, so he's the guy who discovered the Mausner cells in people in zebrafish. They talk about those. So he's the one who discovered that. So he actually proposed another sleep center near the oculomotor nucleus, which is exactly this region, because the third nuclei, they're like here and here, right? So exactly the region that we're talking about. So what's interesting is that when Mausner died, just a few years after he proposed this, the idea of the existence of a sleep center remained highly influential. In fact, von Economou was very much inspired by Mausner's idea of a sleep center. So he went to look for sleep centers, and he found the POA. But the specific center that Mausner proposed near this region was largely forgotten, right? So he was kind of a little bit too much ahead of his time, right? That was his tragedy. But anyway, so when we found this, we were very excited. In fact, I didn't know about this until my postdoc, Zhe Zhang, who found this region, he found some really old reviews that sort of mentioned Mausner's idea. So anyway, so we were excited. On the other hand, even though there are a lot more putative sleep neurons than wake neurons, it's still a very small minority of all the neurons in the ventral pack. So we have to figure out who these neurons are. And to do that, we use gene profiling. So nowadays, sequencing is so much cheaper, so even individual labs can afford that. So what we did here is that now the reporter mouse line that we use expresses not only GFP, but also this ribosomal protein called L10A. So the ribosomes, they're normally attached to mRNA for translation, right? So that's their job. And so if we use an antibody against GFP to pull down L10A, we can also pull down the mRNA attached to the protein, right? So that's the trick. And so once we pull down the mRNA, we can sequence that. So this is what the data look like. I think the people call it the volcano plot. So each dot is a gene, and this horizontal axis is the expression level of that gene in the putative sleep neurons, right, labeled by GFP, relative to this entire chunk of tissue that we cut out for this process. And this y-axis is the p-value. So this red box here indicates the genes that are highly significantly enriched in the putative sleep neurons. So we decided to focus on the neuropeptides, and that's because, one, we know that neuropeptides are important for signaling. Two, we rely on Cree mouse lines that we can buy, right, from commercial sources. And there just happen to be a lot of Cree lines associated with neuropeptides. And so in this box, there's 3 neuropeptides, these red dots. And among them, this peptide calca is a calcitonin gene-related peptide alpha, something like that. Anyway, calca. So this gene, the expression shows a substantial overlap with the GFP-labeled putative sleep neurons. So we decided to focus on these calca cells. So the first thing we did is to make sure that these neurons are truly sleep active, because even though we use CFOS to label that, CFOS is a pretty good but not a perfect indicator of neuroactivity, because it also depends on the cell type and other things. So to really make sure these neurons are sleep active, we did recording. So we used a calca Cree mouse generated by Richard Palmiter's lab. And in this case, we optogenetically tag these neurons by injecting AAV expressing channelrhodopsin in a Cree-dependent manner. So only the calca cells express channelrhodopsin. And then for recording, we use this thing called Optrode. It basically contains a optic fiber surrounded by several bundles of electrodes. So this is something that you can make in-house, right? So it costs for, I don't know, $50 or something. But it does take some practice to make a good one. So here is a recorded unit, right? So this is all in vivo in freely moving mice. So all you don't get to see the cells. All you have is a nice spike, right? So this cell, you can see that every time we turn on the laser briefly, the little blue dot, which is about 5 milliseconds, I think, you see that this neuron always fires a spike reliably at a very short latency. So we're pretty sure that this is a neuron that expresses channelrhodopsin. Therefore, in this mouse, it should be a calca cell. And once we identify a cell like this, we turn off the laser and record a spontaneous activity. So here is the EEG power spectrogram, the EMG trace, and this is the color-coded brain state. So you can see that this cell here fires at a high rate during non-REM, right, the orange periods, and lower rate at both wake, right, gray and blue REM. So here, each line is one identified calca neuron. I think that there are like 33 cells or something. So for these opto recordings, we usually don't have a huge N, right? And that's because it's a pretty darn difficult technique. In some cases, you get 1 cell per week. So if you get it on Monday, you have my permission to just go home and watch a movie or get drunk or whatever, right? So it's a pretty hard experiment. And so we also summarize our data in this 2-dimensional plot. So each dot is a cell. This horizontal axis is the firing rate difference between non-REM sleep and wake, normalized by their sum, and the vertical is between REM and wake. So these identified calca neurons, the blue dots, most of them are on the right-hand side, right? So these neurons are more active during non-REM sleep than wake. but they don't really care about REM versus wake. Now, on the other hand, if you look at the unidentified units, right? So these are the units that are picked up by the same optrons, but they're not driven by laser reliably. So we don't know who they are, right? And if you look at these cells, many of them are on the left. So these neurons are actually wake active. So this tells us that these calca neurons are somewhat special, right? Given the different distribution from these unidentified units. So most of them are actually non-REM active. So this is our first criterion for being a sleep neuron. So next, the question is, do they promote sleep? So this is our standard optogenetic protocol. We turn on the laser for 2 minutes per trial, the blue shading here, and we do that semi-randomly every 10, 15 minutes or so. So here is just a segment of recording with 3 trials, EEG, EMG, and a color-coded brain state. Now, this is a summary of all the trials collected from all the calca mice that we tested. So when we turn on the light at time 0, you can see that there's an immediate increase in the probability of non-REM sleep, right, the yellow line, and decrease in both wake and REM. And when we inactivated these cells using this inhibitory opsin, IC++, which also respond to blue light, but they're chloride channel, then we see decrease in sleep and increase in wakefulness. So based on, so this is bidirectional optogenetic manipulation. And so the other thing that I sort of found when I moved to the sleep field is how much people hate optogenetic experiment. In fact, my very first postdoc I hired to do sleep specifically, she came to my lab for 2 months, and then she went to a summer course in Cold Spring Harbor. And she ran into this very famous sleep researcher, very prominent, I won't mention the name. And then she came back being devastated because she said, she mentioned that she was gonna use optogenetics to study sleep, and this guy told her it's a bad idea. She was like, what do I do? And I said, no, this is great news because it means that we have no competition. Right? We're new and we need to have a little niche for ourselves, right? But anyway, so when we're ready to send the papers out, of course, we run into a lot of resistance. And so we also had to play along and sort of do the experiments they wanted. So they like chemogenetics much better, so we did that. And so it turns out that if we use the GQ mediated chemogenetic activation, we also saw increase in non-REM sleep for about 2 hours after the injection of CNO. And if we use the GI mediated inactivation, we also saw decrease in non-REM sleep. So based on both optogenetic and chemogenetic activation, inactivation, we know that these neurons are non-REM promoting, which is our second criterion. Okay, so as I said, we were trying to find out who these neurons are. We found this calca marker, right? But calca is a peptide, and it turns out that in these subcortical regions, most of the neurons don't just use peptides. They also use a traditional neurotransmitter for signaling, right? So it's almost like the rule. They never use a single thing. And so it turns out that this calca population, about 70% of them are glutamatergic. They express VGLUT2. And the other 30-something, 40% of the cells actually are cholinergic, so they express this CHAT marker. So I won't show you the data, but just tell you that we think that the sleep-promoting effect is caused by this glutamatergic subpopulation. Now, these cholinergic neurons, they're actually interesting. If you activate these cells, you don't put the animal to sleep in any significant way. But what you see is that the pupil basically really become immediately constricted. So we think that these cholinergic neurons are actually parasympathetic preganglionic cells, right? So they're part of the parasympathetic system that control the pupil size. So that's probably also involved in sleep because when we do fall asleep, our pupils are constricted anyway, right? And so some ideas are like, that restricted visual input so that you're less likely to be, awakened by visual stimulation. But they don't promote sleep in a very strong way. Okay, so it turns out that the calca neurons are not the only glutamatergic cells that promote sleep. We also found another population. So these neurons express CCK, which is another neuropeptide. So the CCK neurons are almost 100% glutamatergic. But if you look at the glutamatergic population in this region, only about 50% of them are CCK. And so this just shows that the calca neurons and CCK neurons are totally non-overlap, right? So you see that the CCK cells are the green cells sitting along the midline, and the calca neurons are these red guys sort of straddling that population. And when we activated the CCK cells, we also saw increase in non-REM, right? Inactivation caused a decrease. So the CCK neurons are also non-REM promoting. Okay, so we have these two populations, and they sit right next to each other. The question is, are they just neighbors, or do they ever talk to each other? And so for that, we turned into acute brain slices of the ventral pack. So the trick that we used was to use one Cre line, right? So in this case, it's a CCK Cre, so that we can inject this Cre-dependent AAV expressed channel dolphin into the CCK neurons, right? So you have Cre lines, great. You can target a particular cell type, but you can only target one type at a time. So how do we identify the calca neurons? What we do is that we use whole cell recording, and then after recording, we also suck out the cytoplasm, and then we can kind of do RT-PCR to see if there are, in the cytoplasm, there are RNAs, expressed for that marker. So in this case, we know it's a calca cell. And so you can see that when we activate the CCK neurons, we saw this nice depolarization, and that's blocked by CNQX, right, which is AMPA receptor blocker, so that we know that this excitation is caused by glutamate. And conversely, when we use the calca Cre line to make them the presynaptic cells and record from the CCK cells, we saw similar glutamatergic excitation. So just to summarize this part, right? So I've shown you that in our effort to screen for sleep active neurons, we have found two populations. Both are glutamatergic, calca cells and CCK cells, and they're actually reciprocally connected to each other. So I won't show you the data, but just to tell you that both populations send long-range projections to other regions. Actually, both of them project to the POA, right? So this is the textbook sleep center. There's actually another region in the medulla, very, very posterior part of the brain, right above the spinal cord. So the calca neurons also project to this region called the GIV. So both of these populations also promote non-REM sleep, and both of them are GABAergic. And we also know that they project to a whole bunch of wake-promoting neurons. So it seems like the logic of the circuit is that you have these glutamatergic neurons that are upstream of these GABAergic neurons, right? So these sleep neurons excite these other sleep neurons, which in turn inhibit the wake neurons. So that seems to be a fairly common theme that we found. Okay, so now I'm going to switch to the second approach, which is to screen for sleep-promoting neurons. Now, we did this based on a very simple logic. I'm a pretty simple person, so I need to have a very simple idea so that my brain can handle. So the idea is the following. So we're going to start with the wake neurons, right? So as I mentioned at the beginning, we know a lot of them. And we're going to use retrograde tracing to find their inhibitory input. Because if you're a neuron that inhibits a whole bunch of wake neurons, then perhaps your activity can promote sleep, right? Because sleep and wake, they're kind of antagonistic. And then once we identify the inhibitory sleep neurons, we're going to trace one more step back and look for their excitatory input. Because if you're a neuron that excites a whole bunch of sleep neurons, then perhaps you are a sleep neuron yourself. So the screening was done by Chen-Yan Ma, a very talented postdoc in my lab. So Chen-Yan targeted a whole bunch of wake-promoting neurons, right? There are plenty of them all over the brain. And so she was looking for brain regions with relatively broad GABAergic innervation of these wake neurons. So in the first round of screening, Chen-Yan targeted 7 populations of wake neurons. These include the histaminergic neurons in part of the hypothalamus, noradrenergic neurons in the locus coeruleus, some cholinergic neurons, and some glutamatergic and GABAergic neurons, right, in 7 brain regions. And here's a list of brain regions with relatively broad GABAergic innervation. So she did this using the transsynaptic rabies virus tracing. So among these candidate regions, right, so she found the POA, right, which is the textbook sleep center, which makes sense. But Chen-Yan also found this region called the CEA, which is the central nucleus of the amygdala, right? And so if you just look at this anatomical distribution, CEA actually came even on top, even above the POA. But I'm sure that depends on the specific populations that we targeted. But anyway, so CEA looks like a strong candidate. So just to show you the data, right? So here I'm showing you an example of each of the 7 populations of wake neurons that Tian targeted, right? So that's shown in this diagram, right? So she was injecting 2 viruses in the locus coeruleus or the parabrachial nucleus and so on and so forth. And in each case, if you zoom in to this little region here, this is the central nucleus of the amygdala, and you blow it up, you see a whole bunch of rabies virus labeled GAP expressing cells that overlap with the GABAergic marker GAP1 or 2. So, and then again, we have to figure out who those labeled cells are. And again, we did gene profiling using the same trick that I mentioned to you before. So in this case, in this box here, we found 3 neuropeptides that are highly enriched. And so they include neurotensin, tachykinin 2, and prodynorphin. And when we compared these 3 peptide expression, it turns out that NTS and TAC2 overlap by more than 90%. So it's pretty much the same population. And NTS and PDYN overlap by about 50%. But because NTS is more restricted to the central nucleus, whereas PDYN is like more all over the place, we decided to focus on neurotensin. And so indeed, when we looked at neurotensin, compared that with rabies labeling, we saw a very high percentage of overlap. So it seems like neurotensin is a good marker, candidate marker. So here we tested, we again used the NTS Cree mouse, right, to express channeldoxin specifically. So here's an example experiment, and here's a summary, right? You've seen these plots. So again, we saw increase in non-REM sleep. And inactivation, we saw decrease in non-REM sleep. So the CEA NTS neurons are non-REM promoting, right? So this is the case we were searching for sleep promoting neurons based on anatomy, and functionally we confirmed that they do promote non-REM sleep. And then the same thing, right? Optical recording. So here are the identified NTS neurons, right? So most of them are actually on the upper right quadrant. So these neurons are actually more active during both non-REM and REM compared to wakefulness. And the unidentified units are much more scattered. And so here's just one example. So these CEA NTS neurons are, at least most of them are sleep active. So this is our other criterion. Okay, so like I said, right, so once we find these GABAergic sleep neurons, we're going to trace one more step back and look for their excitatory input. So Chen Yan targeted these CEA NTS neurons and looked for glutamatergic input. So here's one region with strong glutamatergic labeling, right? So these are the rabies labeled cells. Presumably these are presynaptic to the NTS neurons in the CEA. And this is a VGLU2, right, a glutamatergic marker, and you see a lot of overlap. So what is this region here? So this is the Allen mouse atlas. So this red patch here is actually this red region here. It's actually a very posterior part of the thalamus. Unfortunately, people, this part of the thalamus, the atlas would just give them some random names, and if you search the literature, you find very little, right? So people say confusing things, right? So we really don't know what to call this region. But the other thing is that you see that the rabies labeled cells is really this sort of vertical strip, right? Whereas VGLU2 labels this entire sort of like, a big patch, right? So VGLU2 is clearly not a good marker because it labels too many of these syllabic neurons. So again, we have to find a good marker for these rabies labeled cells. Now, in this case, we just made a lucky guess, right? So it turns out that when we found neurotensin for the CEA neurons, neurotensin is actually very interesting. I didn't know this, but every time for me it's common that we'll find some random markers and I Google them, right? And so if you Google neurotensin, it turns out that it's a neuropeptide that was discovered in the 1970s. It was first described as a hypotensive peptide because if you inject it into the body, you lower your blood pressure. Now we also know that it lowers the heart rate and also body temperature, right? All of those things happen during non-REM sleep. So we think that it's too much of a coincidence, right? And so in this case, we were excited about neurotensin, and we said, okay, maybe let's just make a guess. Is neurotensin labeling these cells? And so this is, again, just from the Allen in situ data set. And you see that neurotensin is exactly expressed in this vertical strip rather than this entire patch. And when we compare neurotensin with the rabies labeling, again, we saw a very high percentage of overlap. So here, again, we just use the MTS creline, and then when we express the virus in the thalamic region, again, we saw activation increase in sleep and inactivation decrease in sleep. So again, these thalamic, so we just call it the posterior thalamic neurons because we don't know what to call it, right? So these MTS neurons also promote sleep. Okay, so I just mentioned that MTS is an interesting peptide, and now we found that it actually labels two populations of cells, right, in this pathway, right? And one is glutamatergic and the other is GABAergic. So, I was giving this talk for actually quite a while, and the paper only came out last year, right? It took us almost 2 years from the first submission to the time the paper was finally accepted. Anyway, so during this period, I was giving this talk, and people always ask me, what's the function of MTS? So initially I said, I don't care, I'm not a peptide person, all I care about is a creline. Once I have that creline, I label the cells and it's done, right? But when you ask that question like 20 times, you get sick of your own answer, right? So finally we did experiment and tested this. So what Chen-Yan did was to design this very complicated looking construct. I don't know how she designed it. She's a genius in molecular biology. But anyway, so the important thing here is that it actually expresses this guide RNA against MTS gene, right? So then if you inject this into this complicated mouse configuration, right, which is MTS-CRE, but also express Cas9, right? So you can use the CRISPR-Cas9 technology to kind of manipulate the gene expression. So in this case, basically we guide the Cas9 to kind of clip the MTS gene so that the expression is knocked down. So as you can see here, right, this is the control mouse. So she, again, expressed something like this, except that the guide RNA is not against MTS, it's against some random gene. So that doesn't affect MTS expression. But if you express guide RNA against MTS, then you really knock down MTS expression. So that's true for the CEA and also for the posterior thalamus. Now, when we tested the effect of optogenetic activation of either population, so this bar is the control, right? So this shows that, so this is showing the effect of optogenetic activation, how much increase in non-REM sleep we see, right? So you see this nice increase. Now, if you knock down NTS, then the effect is greatly reduced. But if you knock down this vesicular transporter for GABA, it also greatly reduces the effect of activating these cells. So this is true for the CEA. Now, in the posterior thalamus, if you knock down NTS or knock down the vesicular transporter for glutamate, both of them really pretty much wiped out the non-REM promoting effect. So the conclusion is that basically both the NTS peptide and the traditional transmitter, right? In one case is glutamate, the other case is GABA. They're all required for the sleep promoting effect. But it turns out that these are not the only two NTS population that promote non-REM sleep. So now we figured out, NTS is interesting. We just got lazy, right? So we just and said, okay, where else is NTS expressed? And you go to the Allen in situ data set, and so that shows that NTS is also expressed in the PAC as well. And when we activated these PAC NTS neurons, we also saw a huge increase in non-REM sleep. And inactivation, a clear reduction, right? So here's a third population of NTS expressing cells that promote non-REM sleep. So Peng also did optical recording, but here just for a change, I'm gonna show you a different technique that we used. So in this case, it's calcium imaging, right? So we can express GCAM6, again, using NTS creline to express an NTS cell specifically. So in this case, because most of the brain regions that we're looking at are subcortical, they're buried deep in the brain, so we have to use a special thing to be able to image them. So in this case, it's a green lens, the microendoscope. And then for recording, we use this miniaturized microscope developed by Mark Schnitzer's group. So it's so tiny so that the mouse can actually wear on the head and still move in the home cage. So here, you can see several cells in the field of view, and here's the calcium activity, and basically, again, we saw that these cells are more active during non-REM sleep and less active during the other states. And so this data is also confirmed by some output recording. And so again, for the PAG NTS cells, we also knock down MTS expression. And so this is the control, right? Nice increase in non-REM sleep. You knock it down, NTS, the effect is reduced. Or if you knock down VGLU2, right? So this is glutamatergic transmission, the effect is also greatly reduced. Okay, so just to summarize this, right? So we have found NTS neurons in the CEA. These are GABAergic, posterior thalamus, glutamatergic, and also the ventral PAG, also glutamatergic. So all of these promote non-REM sleep. And in all of these cases, both the peptide and the traditional transmitters are important for the sleep promoting effect. Okay, so there are two other points I kind of want to emphasize. So one of them is that NTS seems to be a really general marker for multiple non-REM promoting populations. So I showed you these three. I haven't shown you the data, but there's actually a fourth population in the subthalamic nucleus, which I'll talk about next. And so the second point is that it seems like the ventral region of the PAG really is an upstream excitatory sleep center, right? I told you about CCK, CALCA, and also NTS. And they send broad projections to a whole bunch of GABAergic sleep-promoting regions. All of them, in turn, inhibit the wake neurons. Okay, so now in the remaining time, I just want to quickly take you through the last study actually that Reid mentioned. So this region was also came out of this sort of screening, right? So I've told you about CEA and the POA, right? So the third region based on this anatomical screening is the SNR, the substantia nigra pars reticulata. And so this population was characterized by Dan-Qian Liu, another incredibly talented postdoc in the lab. So now some of you might know that the SNR is actually part of the basal ganglia, right? So that's an important part of the somatic motor control circuit. So to get an idea of how these apparent motor neurons might also be involved in sleep control, we decided to take a closer look at the motor behavior of the mice. So in addition to EEG and EMG, we also added video recording. And as some of you might know, deep learning is all the rage nowadays. Almost the whole planet is doing deep learning. So we didn't want to be left out, and that's why we got into it as well. So our collaborators in Beijing, in Hua Han's lab, they're the expert in that. And so they helped us to use deep learning to train a convolutional neural network called UNet. So this is for image segmentation, right? So this is the original image, and this is where the network thinks the mouse is. So once we can automate this, now before this, we do it and we just get a whole bunch of undergrads. We have a lot of undergrads in Berkeley, so they sit there and click, right? So, running, okay, eating, grooming. So now we can actually do this in an automatic way. And so it's pretty straightforward once you have a large data set to extract some simple parameters. So the two parameters that we extracted were pretty kind of, like intuitive, right? So one is translation, right? So we look at the center of mass of the mouse in two different frames and see how much it moved. The other is total movement, right? So basically we look at the total number of pixels that are not shared between these two frames, right? Because sometimes they don't really run, but they might change their position a little bit. And when we plotted these two parameters, we saw three clusters. The dark green cluster corresponds to local motion. The light green corresponds to all the non-local motor movement. So we specifically decided not to do a very fine-grained classification, right? So non-local motor movement includes a whole bunch of things, including grooming, right? So they do this, like clean their whiskers a lot, or eating, right? Or just some postural adjustment. And the third cluster basically corresponds to immobility. But when we looked at the EEG and EMG in this cluster, there are actually two different states. One is quiet wakefulness, right? So they're sitting there, not moving, but their EEG and EMG shows that they're really not asleep either. The other is sleep. So in this case, we just lumped REM and non-REM sleep together. So here I'm showing you the EEG delta power across these 4 behavior states. So the higher the delta, which is low frequency activity, the less aroused the brain is, right? So when you're really active, you have very little delta power. And then this is showing the total EMG power. So the point I want to make here is that now if you look at these 3 green states, they're all wake states, right? So different movement or quiet wake. So we tend to think of their difference in terms of motor activity, right? On the other hand, if you look at their EEG delta power, there's also a clear difference. Now, both quiet wake and sleep, they're immobility states, right? So we tend to think of their difference in terms of the brain state. On the other hand, if you look at the EMG, there's also a pretty clear difference. So the point is that brain state, as reflected by EEG and motor activity, they're correlated across multiple behavior states, right? It's just that somehow people in different fields tend to focus on different aspects, but behavior is all, is all the same. So when we looked at the transitions between these states, they're not random at all, right? So if they start out in local motion, they always transition into non-locomotor movement. We have never seen the case where the mouse was like running one moment and bam, fall asleep next moment, right? If you think about it, we don't do that, right? So I have to say that this is true for normal mice, but if you have a narcoleptic animal, they would do that, right? You might have seen some YouTube videos. There's a guy called Rusty, the narcoleptic dog, right? It's really funny. He would like leap, right? And then, right in the middle of the air, he's like fall asleep and bam, he falls down and he's like eyes are closed and he's asleep, right? So, but that's abnormal. So for normal mice, we don't see that. Now, if you start out in non-locomotor movement, you go back to running or you go into quiet wakefulness and you don't go to sleep right away. So if you place these 4 states in a single chain, then most of the transitions are between neighboring states. So here's a transition matrix. Now, diagonal just means from one moment to the next, they're staying in the same state, which happens a lot. Right above the diagonal, these are moving down the chain one step at a time, and these below the diagonal, they're moving up the chain. So the only exception that we saw is that sometimes they wake up from sleep and they go right into movement and skip quiet wakefulness, right? So that's the only non-neighboring state transition that we saw. And it actually happens at a pretty low frequency. Okay, right, so this is all just behavior, right? So back to neuroscience. And so we were gonna talk about the SNR, right? So it turns out that there are 2 subtypes of GABAergic neurons in the SNR. If we inject this Cre-dependent virus into the parvalbumin Cre mice, we mostly label the PV positive cells in a lateral SNR. But if we inject the virus into GAT2 Cre mice, we mostly label the medial SNR. And so these cells are actually PV negative. And you can also see that there's a fairly clear spatial segregation between the 2 cell types. So here I'm showing you the output recording from the PV cells. Here's one example, right? So here I'm just color coding the behavior states. Other than just sleep and wake, I'm also color coding the different motor states. Now here, each line is 1 PV cell, right? And so you can see that most of the PV cells are more active during these movement states and less active during sleep. So these cells are clearly not sleep active. But when we did the same thing for the GAT2 cells, most of them are most active during sleep and less active during these motor states, right? So these cells are actually sleep active. So here I'm just showing you the superposition of the population average of the 2 populations. So when we activated the GAT2 neurons, so again, it's the same kind of thing, right? So laser starts at time 0, right? It's blue shading. So we saw an immediate, pretty much wipes out locomotion and also strong decrease in movement and a clear increase in sleep. But when we activated the PV cells, we basically saw no effect on sleep. The main effect that we saw was a decrease in movement and increase in quiet wakefulness. So they mostly transition from some kind of movement to stop moving. And so here's just a bar summary of the same thing. And when we inactivated the GAT2 neurons, we saw huge increase in movement and decrease in sleep. and the PV neuron inactivation has much weaker effect. So a quick summary, right? So the PV cells are actually movement activated, right? So remember, they're mostly active during movement, but their activation mainly terminates movement. So we think what's going on is that the PV neurons are important for action selection. So this is from the work from people who study the basal ganglia, right? So there's this idea that every particular movement you're making, you also have to suppress these other competing movement, right? So which is why whenever you're making any movement, there are always some PV cells that are suppressing these other movements. And that's why overall they're movement activated. But when you artificially activate them, you're mostly terminating the movement because you're activating the entire population. But the GAT2 neurons are different, right? So the overall effect is to suppress all movement and actually increase sleep. Okay, so even though the GAT2 neurons really suppress movement and increase sleep, they don't cause direct movement to sleep transition, which is not normally observed, right? Instead, what they do is to bias the direction of the natural transitions. So here I'm plotting the change in the transitions caused by GAT2 neuron activation. So you see that these zero transitions are still zero, right? But for the existing transitions, you see that these guys above the diagonal, they're all increased, meaning the transitions going down the chain is enhanced, but moving in the opposite direction, they're suppressed. So we think that this actually provides some answer as to how the same population of neurons can both do motor control and sleep control. And that's because, when we look at this transition, going from running to other movement, we say, hey, that's movement control, right? If you talk to a motor researcher, they will say, any change in movement.
Before you mentioned the PV neurons function, is this PV neuron positive or negative?
Oh, so these are all GABAergic. These are all GABAergic.
PV neuron is positive V-glutamate and negative. Sometimes some areas negative V-glutamate.
Right. So as far as I can tell, in the SNR, there are very few glutamatergic neurons. I don't think my lab directly compared the PV with VGLU2, but if you look at the ALAN ALICE, you see very, very few glutamatergic cells, I think. Yeah.
Do you know anything about where this subpopulation of SNR cells project?
I'll show you.
Okay.
The next slide, I think. Okay. So when we look at this transition, we say it's motor control, right? And then the next transition is also motor control. But this next transition we say is brain state control, right? So we call them different things. But in fact, these neurons, they really don't care what we call these, right? Basically, all they do is to move the animal down the chain, right? And for each transition, there are actually both EEG and EMG changes that I showed you earlier. So I think that this sort of motor control versus sleep control is really artificial. It came from us, right? And so here, if we inactivate them, we saw the opposite, right? So all the downward transitions are decreased and the other transitions are increased. Okay, so here's the answer to your question. So we looked at the downstream target, right? So if you look at the PV cells, they project to the motor part of the thalamus, the motor layers of the superior colliculus, and also this region, I think that the motor guys call this the midbrain local motor region because it's really important for local motor control. So the GAT2 neurons, they project to these motor regions, but in addition, they also project to the dorsal raphe. So this is a region that contains both serotonergic and dopaminergic neurons. And so people have shown that those neurons are important for arousal and wakefulness. The GAT2 neurons also project to the locus coeruleus, right, containing noradrenergic neurons, also important for arousal. So when we label the subset of the GAT2 neurons that project to the thalamus, right, so these are thought to be the motor control neurons. In fact, we also saw their axon collaterals in the dorsal raphe and also the locus coeruleus. And when we label the subset of neurons projecting to the dorsal raphe, we also saw their axon collaterals in these motor control regions. So it really seems like it's the same population of neurons that sort of send axon collaterals to these multiple regions, right, including both the motor control regions and the sort of arousal regions. Okay, so again, we found a GABAergic population and we want to find their glutamatergic input. And it turns out that, of course, for people studying basal ganglia, they all knew this, right? But except for us, we had to find out with rabies tracing. So the strongest input to SNR came from the subthalamic nucleus. I think that some of you guys study here, right? So these neurons, they're glutamatergic, and they also happen to be neurotensin positive, right? Which is what makes neurotensin even more interesting, right? They're also in this somatic control region. And so when we activated these NTS expressing neurons, just because NTS is more specific in the STN, VGLU2 is too broad. When we activate these cells, we also saw an increase in sleep and decrease in wakefulness. Okay, so at the beginning, I told you that falling asleep is associated with a dramatic change in brain state, and also reduction of somatic motor activity, and also the autonomic motor output. So it kind of makes sense for the sleep-promoting neurons or the sleep center, whatever, to inhibit the arousal system and also these two types of motor systems. So I told you just now that the STN, SNR, right, the circuit strongly promote sleep, and they're part of the basal ganglia somatic motor control circuit. Now, I also told you we have found neurons in the periaqueductal gray, the PAC, right, the amygdala, the hypothalamus, and also some data in the nucleus of the solitary tract, which I didn't have time to show you today. So it turns out that all of these regions are important nodes in the central autonomic network. So instead of being separate, as implied by this circuit diagram, it seems that the sleep control circuit deeply infiltrates both the somatic and autonomic motor control system. So that's why I moved these yellow sleep neurons into these two motor boxes. On the other hand, the general state neurons might be separate from the specific action neurons. So what I mean is that in the SNR, the PV neurons seem to be important for action selection, but the GAT2 neurons are the general state neurons. And these state neurons are the ones that send axon collaterals to both the motor control neurons and also the arousal system, right, including dorsal raphe and locus coeruleus. So we think that this kind of view of how sleep is controlled also has some implication for the question of why do we sleep, right? So everything I talked about today is about how we sleep instead of why we sleep, which is even more fascinating, right? So this is something that I only started thinking about for about a year or so, and it's probably going to be the focus of our future research. So the fact that these sleep control neurons are so closely associated with these motor control circuit suggests to me that sleep is not just about changing the brain state, right? It's equally important about suppressing these motor output, probably to promote processes incompatible with movement. I don't know what that process is. I have some guesses, right? And so we're doing some experiments, and who knows, maybe 3 months later, I don't believe a thing I'm thinking about now. But I think that it's important to remember it's not just about the brain, it's also about the peripheral, the whole body. And part of the reason to think that is that if you don't sleep well, you don't just have cognitive impairment, you also have many other impairment in the body, immune system, cardiovascular, and so on and so forth. So finally, these are the people in my lab who actually did all the work. So Zhe Zhang is the one who did the screening for sleep-active neurons. Chen Yan did the screening for sleep-promoting neurons, and Dan Qian studied the SNR, STN pathway. And so Peng discovered the NTS neurons in the PAC. So these three people are the early pioneers in my lab. So Xinjie is the one that I hired 9 years ago. She's the very first person who came to my lab to study sleep only. And so these three people really got a lot of the key techniques to work in my lab. So without their effort, we wouldn't be where we are. And all of them are running their own labs now, and Zhe also. And these three are currently on the job market. And we also have many, many collaborators who helped us with many techniques. And thank you.
Time for 4 minutes questions. 1, 2.
So at the very end, you said something that I was thinking about since the beginning of your talk, and I just asked the question. You talked about it's not just the brain and the state, it's the body and the nutrition. So I kept looking in the exquisite elaboration of that circuitry for potential sources of feedback from the body in terms of afferent signals from whatever, proprioception, et cetera. Where might that come in and how important?
Yeah, it's a great question. So I only have some guesses, right? So I think a lot of that kind of signal is probably not the somatic sensory signal. It's probably the visceral kind of input, right? So the somatic system has a somatic input and the somatic motor output. And the autonomic nervous system, we say, is a motor system, but there's also sensory input. So the visceral input, a lot of them actually, the afferents come into the nucleus of the solitary tract. So I didn't show you any data there, but I sort of in the summary plot, I showed that that's one of the autonomic central network. So that's actually the sensory stage. And in fact, we have found some sleep neurons there as well. And they're actually specifically involved in cardiovascular regulation.
Yeah. All right, thanks.
So one thing I'm wondering about is how this circuit gets engaged. And one obvious thing is circadian rhythm and keeping track of those sorts of things. And so I'm wondering just generally how you think about the interface of that. Then I have a specific question, which is that for the calca neurons, one population projects to both the GIV and the POA and another doesn't. And do you think that that has any significance for sort of balancing their ability to activate sleep?
Yeah, I mean, the second question is easy to answer. I don't know. It's interesting, right? But it's pretty clean. One population clearly projected both almost equally. The other, like you find no axon projected.
That's really interesting.
Exactly,
That sharp division.
Exactly. So I really don't know. There's got to be some clean logic. Because sometimes you find, 70% this way, 30%, or the other population like 40, 60, or like whatever, right? But this is a very clean effect.
Yeah.
There's got to be something, but we don't know yet.
Okay.
The first question, what we do is to sort of find the core sleep circuit, right? In the sense that you activate those neurons within 10 seconds, you see an increase in sleep, right? But there's this famous two-process model proposed by Borbell or something. I'm pretty sure I'm pronouncing that name wrong. So anyway, he proposes two-process model. The idea is that sleep is regulated by two things. One is circadian clock. The other is homeostasis, right? In the sense that if you stay up for 20 hours, you feel sleepy no matter what time of the day, right? So we looked into the circadian regulation a little bit, and then I kind of just gave it up. So what we found is that if you activate some neurons in the SCN, the VIP neurons, I believe, what we saw is it doesn't quite make sense. So we specifically saw an increase in REM sleep, not non-REM. You would have thought that, okay, so mice are nocturnal, so if you activate the SCN, which is active during the day, you should increase sleep overall. But we only saw increase in REM. So I could never figure out how to explain that, right? And then we had a hard time trying to figure out the downstream target and all that, so we just kind of put it away. And also because the circadian field is very mature. So it's kind of hard for someone new to jump into it and do the right thing, right? So I decided to just close off that direction. But we are very actively looking at sleep homeostasis. And that's because, as I said, I'm interested in sleep function, and there's got to be a very tight relationship between homeostasis and function, right? Because if sleep is important for process A, then when you need process A, you should feel sleepy, right? So I think that that's sort of a tighter, it's probably easier to do, and also scientifically it's a more compact problem.
Great talk, really excellent. I just had a couple of questions. So, of course, mice are quadrupedal and humans are bipedal. So I was wondering if there may be additional nodes or firing rate encoding involved in moving from the standing position to the laying position. Obviously you lay down when you sleep, but we can also sleep sitting up, which involves some possible control. And then the second naive question was there's a variety of species which have locomotion through fluids, like dolphins and some species of bird, which can actually sleep while maintaining locomotion. So I was wondering,
Yeah.
what you thought about that and how that fits into your idea of why.
Yeah. So again, I'll answer the second one first because I remember it better. So so I talked to this guy Rottenberg, I think. So he's actually American, but living in Berlin, I think. So he actually found these the what's it called? Basically sleeping half of the brain at a time. I'm just stuck on that word, right? Yeah, right. So we were at a sleep symposium in Paris just a few months ago, and so one question is always, when the bird is flying, right? So on one hand, the two sides of the brain take turns, right? On the other hand, what's happening with their motor system? And so I was actually nervous, right? Because this is something I don't know because I can't possibly study birds in my lab, right? But then it could be a problem. And then so I was really worried that he was going to give me a hard time. And then I didn't know the answer, right? But it turns out he actually liked it because when he studied the behavior of the birds, right? When they're sleeping, what they do is that they first sort of find some air current, right? And so they actually sleep when the current can kind of carry them for a while. And they sleep with very short bouts. So that is a little bit sort of anecdotal, right? Because it's actually hard to characterize that when they're flying, right? But that's sort of what he thinks is going on. So there, at least so far, there doesn't seem to be a big problem with this sort of shutting off motor control and that, right? So your first question, so I'm going to turn into a question that I know a little bit more about. So when I gave this talk in Israel a year ago, this guy Hagai Bergman, so he's a leader in basal ganglia research, and so one of the things he told me is that rodents have a lot more sort of a postural control. So their basal ganglia, a big part of it is sort of devoted to the trunk, right, to the posture control. Whereas we primates, we have a lot more sort of, we do these menu things, right? So the lateral part, so there's also medial lateral gradient. The medial SNR is more like for the trunk, and then the lateral is more for this, right? And so I think that there's some relationship with the PV GAT2 division, right? So if you read the SNR literature, which is actually not that large because a lot of the studies in basal ganglia is more the striatum stuff. But in the SNR, people talk about the lateral SNR being important for sensory motor control. The medial part, they call it the association area. And so in my mind, every time you call a brain region association area, it just means you don't know what it's doing, right? So that's sort of my current view of related to that.
Okay, well, I'm going to call it there. Thanks again for coming.